Hello, 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 hello. Yeah. A very good afternoon to everyone present here. I take this privilege to warmly welcome all our distinguished guests, colleagues, friends from various departments, institutes to first Professor Tikaram Sharma Memorial Lecture and World Down Syndrome Day organized by Center for Genetic Disorders, BHU Varanasi. A very special thanks to all of you for gracing this occasion with your esteemed presence. This lecture is the first one in a new series to honor a distinguished academician and legend, Professor Tikaram Sharma, who contributed to teaching and research in cytogenetics immensely. His contribution mainly covers radiation-induced aberration and heterochromatin evolution. This day marks the celebration of his greater visibility as a scientist, teacher, and mentor. As is the custom of BHU, we will start this program with the Kulgi. For this, I request Prashasti and team to join me. And for a little information for the new students, please do not clap after Kulgi. मधुर मनोहर अतीव सुंदर ये सर्व विद्या की राजधानी राजधानी सुरम्य धारा के बनुरासी नहाए जिनमें कबीर तुलसी भला हो कविता कपियों लाकर ये बाग बेटिया की राजधानी राजधानी मधुर मनोहर अतीव सुंदर ये सर्व विद्या की राजधानी राजधानी विविध कला अर्थशास्त्र गायन गणित खनिज औषधि रसायन नतीज प्राचीन मेल सुंदर ये विश्वविद्या की राजधानी राजधानी मधुर मनोहर अतीव सुंदर ये सर्व विद्या की राजधानी राजधानी ये मालवी जी की देशभक्ति ये उनका साहस शक्ति प्रकट हुई है नवीन होकर ये कर्मवीरों राजधानी राजधानी मधुर मनोहर अतीव सुंदर ये सर्व विद्या की राजधानी मधुर मनोहर अतीव सुंदर ये सर्व विद्या की राजधानी राजधानी Thank you, Prashasti and team. Now I request our coordinator, Sir, Professor Parimal Das, to come on the stage and give welcome address to the gathering.
Honorable Director, Professor A.K.Tripathi, our guest of honor, Dr. Tulsi from DISCC, the DEVA International Society for Child Care, our keynote speaker, Professor Rajiva Raman, our distinguished guest, Viva Mishra. She's also a member of our Human Ethics Committee, senior teachers, Professor Lakhotia, Professor B.N. Singh, Professor Mercy Jacob Raman, and Dr. Sukala Prasad, other colleagues, aides, students, non-teaching staffs. On behalf of myself and on behalf of Center for Genetic Disorder, I welcome you all to join today's gathering, which is especially meant for Professor Tikaram Sharma's first memorial lecture, along with Down Syndrome Day, which was this year, was a World Down Syndrome Day, it's generally on 21st of March. But it is a good thing we tried to club this together and we're done. Also, I would like to warmly welcome all the distinguished personnel, family members who have joined us online. I want to mention about Madam Sharma and the family, and then Dr. A.K.Tripathi, then I think I can see on the screen Dr. Awadhesh Jha, Dr. Bhudev.C.Das, Satyan Maitra, and many others who were very warmly reciprocated for their presence. Professor Madan Mohan Chaturvedi was over here and in the Delhi University. So I welcome you all for this. Those who have joined us either online or offline for the first time, I would like to say a few words for Center for Genetic Disorders. By seeing today's gathering and with the motto of ours, I can see that it was deeply rooted with Professor Sharma's thought who was long back, along with the members at that time, thought of using chromosomal diagnosis for the disorders. So I think probably we are trying to do something what he thought long back. Center for Genetic Disorders started established on 2006. We do run a chromosomal genetic and molecular diagnostic unit since long. It was started by Professor Rajiva Raman, and it was carried over here that we are trying to give and service to the society. We also run a postgraduate diploma course on chromosomal genetic and molecular diagnostic to make highly skilled personnel who can go directly for challenging job like genetic counselings and then working in the company R&Ds or higher education. Faculty member, we also run a PhD program along with human molecular genetics. We have started long back, and then already we have 10 students who have already done their PhD, and then they are engaged in higher order studies. And currently also we have like 13 PhD students, faculty members of CGD are involved in various research in human genetics. We have also been connected with the research and consortium internationally and then nationally as well, like faculty members from CGD has been invited to take part in a global consortium on Parkinson disease, which is run by MJFF, Michael J. Fox Foundation, where we are taking part as a invited member where you find out 123 scientists across the world, and they are coming from like 83 different institutes. Also, recently, Department of Biotechnology started a UMED program, which is actually stands for Unique Method for Management and Treatment of the Inherited Disorders. We are also contributing over there. We are taking part, faculty members from CGD and IMS, PHU are taking part of that. And also very recently by DBT's program for pediatric rare genetic disorders. From here, myself and from IMS, Ashok Kumar sir is also taking part in that. Faculty of CGD also running a human ethics committee over here. This is the Nodal Center for. This is the in short for genetics. What I can say and can see and then promise that we are trying our level best to go as Professor Sharma also thought long back. Now, with these small words, again, I welcome you all for today's matter. Thank you.
I also request Sir to please present the bouquets to our guest of honor, Dr. Tulsi, as well as our Director, Institute of Science, Professor A.K.Tripathi. The key to success to this lecture series entails dissemination of knowledge. In every sphere of our life, we follow some basic tenets or principles. For these basic tenets, we owe to great academicians and practitioners. Professor Tikaram Sharma was a teacher par excellence. Since this lecture series has been instituted for a worthy purpose, I invite Professor Mercy Raman, former head Department of Zoology BHU, who had a chance to work in close association with Professor Sharma during her PhD days, to give us an insight on the great scientist's life and his achievements. I welcome you, ma'am.
Good evening, everyone. I am very happy to see Mrs. Sharma online. Namaste, madam. Today is the day we always used to call sir, wishing him birthday. And I am happy you and family could be with us on this day when we are together, remembering him and recollecting the great achievements he made in BHU. Distinguished guests, participants online and offline, all the teachers, students, research scholars, everyone in the group. Well, I am not having the fortune of being taught by Professor Tikaram Sharma in a classroom, because when I joined that year, he did not teach our batch, though very short time he appeared for some practicals in meiosis. That too, along with his colleague, Mrs. Ranjiti, and she was attending with the girls. So I lost the opportunity to be his classroom student. But later on, when I joined Professor Raichaudhuri for research, I was in the same lab where he had then been appointed as lecturer and one of the senior most person there. Well, he had joined Department of Zoology along with Professor Raichaudhuri, who was invited to become head of the department and to organize this institution. He had joined Professor Raichaudhuri in Calcutta for his research on grasshopper meiotic damages with radiation. And Sir had asked him to stay on there and continue working as there is nothing in BHU at that moment. But his enthusiasm to be with Sir and to help him, along with two other students, he moved on here. And most of the time, he was instrumental in running about, anybody in BHU, how much running about we had to do between engineer's office, PWD, and all those sections. So his time was totally devoted to developing this department, the structure of it, while Professor Raichaudhuri was busy with organizing the academic front of it. So his PhD time prolonged, and we all have seen him from a distance, and anybody, and most of you here would have seen him, met him, known him. So through our experience, anybody who will see him first, you get a charge of waves, vibes of energy, very pleasant and disposition, very jovial, very friendly. That's what any student would feel. He or she won't feel that he is something to be kept on a pedestal, because he will come down and meet you. He will be one of you, your friend. So when we were there in the lab, so we used to feel that he is a great man, but still we never felt that we could not go and talk to him. That distance was not there. He was not on a pedestal. That was his quality. He was with us all the time. So there, the importance of all this was that he was my Guru Bhai, because I was one of the last PhD of Professor Raichaudhuri in this cytogenetics lab, and he was one of the first among the three. So I used to feel that I have a special bond with him. Being Guru Bhai, everybody in BHU know the Guru Bhai. So we, in the lab, I was in the genetics section, Drosophila genetics. He was heading the cytogenetics section. Occasionally, we used to go there, meet, and have tea, all these things together. So then he will come down and he will speak in a very friendly way. So we used to think that this great man doesn't keep a distance, so we can be talking anything to him. We can go and discuss science, anything other also, any other matter. So that's how we used to see Professor Sharma. Later on, when I became a staff member, and then he's grooming, making me a good teacher, how to be a good researcher, all these contributions I think now. Because in the lab, those of you know cytogenetics lab, you know that there are no boundaries, there is no lock and key. All these things was introduced by him, because he kept it as one family. Why? He came from a big joint family where everybody was caring for everybody, and responsibility was shared, love was shared. That feeling he continued in the lab. So he always cared for the elder people, and then his ability to make friends in the scientific community in India and abroad. We used to watch with wonder how he can easily make friendship with scientists. So we had labs in Delhi University, Professor S.R.V. Rao, and Calcutta University, Arvind Mukherjee, then Gujarat University, Dr. V.C. Shah. All these people come and they all sit together, and there is no curtain, no, that nowadays you may not see the professors are sitting and the students are away, only bringing tea and like that. No, we are all sitting around and we hear their talk and we learn a lot of things. That is a cohesive family unit like thing. So there is nothing to hide because all the discussions are going on along with us. So we grew that way. And then he had very good, few things about him. If I have a lot of time, I could say, but very little time. One thing, first thing and foremost, he was an institution builder. As I told you, that Department of Zoology initial development, many of you may not know, in early years, it was working in the physics department wing. It is Professor Raichuri who constructed this building. So Professor Sharma was left and right hand of him in developing this. And so he designed how, where it should be, how it should be. So that is one thing. Then the cytogenetics lab. So everything about it, he was in charge, and the way he wanted it to develop. He was the first person to go and learn tissue culture, that is lymphocyte culture. So in our country, there were not many laboratories where tissue culture was being done. So he got training from abroad. He was very enthusiastic to establish a tissue culture lab, and he transferred all the knowledge, and the successful culture came out. The karyotyping was done. So initiation of this aspect. Then his two loves, one was area of evolution, and in that specifically how chromosomal changes would bring about evolutionary changes. And later on, his interest concentrated on heterochromatin. Heterochromatin is supposed to be inactive, and how the increase in decay for heterochromatin regions can make changes in evolution. This became a very important aspect of his work. Then another aspect he continued was radiation genetics. So different also chemical and radiation effects, how it will make chromosomal aberrations and mutations in the different organisms chromosomes. In this aspect, he went, so in both the aspects, finally he could achieve his dreams. His studies on heterochromatin particular regions, the non-coding regions, how its changes were how male and female um, deers have differential radiosensitivity. The male is very more radiosensitive. So such path-breaking discoveries were made in both the fields, evolution field and in cytogenetics field. So with this, he carried out different conferences. With radiation biology, he was considered to be an expert in radiation biology, and so he was offered to join an international project by International Atomic Energy Commission, and 6-7 countries participated, and he was the only participant from India. So we had multicentric project, international project, for establishing the low radiation risk. So Awadhesh Jha did wonderful work in that area to show how even a person operating in a that clinics where X-ray machines are operated, such technicians also how they have got radiation. So his vision in different fields brought out through different students' work. It is not that like today's students that every day you ask what did you do and what did you get. Professor Sharma will suggest a problem, and it is for the student to work out and carry out. So he used to say, I am putting you into swimming pool. You know,
Chechi, you're muted. You're not audible, Chechi.
muted.
Yeah, and now
Michelle,
it's fine.
am I audible now?
Yes, yes.
So you missed all the talk? Okay, just that's okay. So this kind of contributions made him an international figure. When Chernobyl accident took place, then he was the person from India to be invited for going and measuring and knowing the details of the damage. So now again Chernobyl is in news. So I was recollecting how he was going and measuring all the damage. So Professor Karl Sperling, who may be on the online line, so his friendship with so many international scientists. So they had first name calling, Tika they will call, and then Nat, Natarajan. So they were close friends, Professor Karl Sperling, and then so many others like that from different laboratories abroad. So science, especially the experimental science, is never an individual science. It is a teamwork. So, so making a network of Indian scientists and foreign scientists who are doing cooperation and doing that is excellent work. When Bhopal tragedy happened, then some of us went and did the initial work of whether the gas has any effect. So later on, that attracted him, and then an ICMR multicentric project was granted. So in that, our laboratory was very active. We prepared the protocol to go there. So in Gandhi Medical College, we made a base lab where they did not know anything. We went and taught them how to do culture of chromosome preparations and analysis. So all these works, so different works as this. So institution building like this continued when he was as a senior reader here. He was selected by government to go and establish a life science center in North Bengal University. He accepted the challenge. He went there, I think, for 5 years. He worked there, made new buildings, and then he developed the departments, made appointments, and then it was time to come back to BHU. He continued here as a professor and worked like that. So, on any aspect, if you think that it's not only science, we were taught how to enjoy doing conferences. The way he would teach us how to become a volunteer, how to do things, he will never impose on you. You have been given a section, okay, you manage it. Only finally there will be a meeting to know that what you have done, and never any reprimand. When we were doing an international symposium on DNA repair, lots of foreign dignitaries had come. I don't think so far we had seen such a big gathering of international people anywhere in BHU. And that time I did a mistake. I forgot to include the name of the DRDO Life Science Director, Professor V.K.J. Professor Sharma would have been wild. Such an important person has come. He is asking, where is my room? And it was my mistake. And he took it on himself. And he said, okay. Then he later on told me, Mercy, you slipped somewhere. That time new ways of computer had come, and then handwritten. We were only used to handwriting notes. And so suddenly transferring into computer notes, somehow the name got missed. So I was so feeling so bad that in front of Dr. V.K. Jain, he accepted it as his mistake. So all good things we are credited. All bad things he will take the responsibility. These were all good lessons to learn from him. And then anything beautiful like gardening. He taught me how to do gardening. When our house was being constructed, he came and told that here will be your lawn, here will be a rose garden. So when people come and see that you have a lawn at the back or side, people make lawn in front. Then I remember, he said lawn is a private place. Like a British people, you will share your private moments in the lawn on the side. And roses, he told me the names of roses, how to grow them. So gardening. Anytime we could discuss international politics, anytime we could discuss cinema, theater, everything with him. So a wonderful, complete personality. And those who could not meet him, I think you miss him. And when he retired and went off from here, I don't know, there was such a vacuum in our life. We didn't know how to live without him. He was a great guru, a mentor, and we were feeling his absence greatly. Of course, he used to come down in between, and sometimes, many times, he used to have long talks. And Mrs. Sharma, I should say, a wonderful hostess, a very kind lady who will take care of all of us like her own children. So, um, I can't say anything more about him. So time is limited. I think I have exceeded my time, but it is an emotional moment. Lastly, I want to congratulate Professor Parimal and his team for thinking of establishing this memorial lecture series, and which is highly deserved. And though with lot of ideas he has come up and associated with the Down Syndrome Day. So two things are coupled together, and we wish this center a great future and hope they will continue with this program always. Thank you very much.
Ma'am just walked us down the lane of blissful memory of Professor Sharma. Thank you, ma'am, for your wonderful reflection on Professor Sharma's life as an institution builder, mentor, and as a person. His life is an inspiration to us all. We are fortunate enough to get in contact with Professor Sharma's family, who have sent us a very beautiful and emotional video, which we would like to play for all of you. Sorry for the delay.
Is ma'am muted? Ma'am, are you muted?
No, ma'am, you are audible. We can hear you, ma'am.
Not me, ma'am. Vishnu ma'am.
We are not hearing the audio.
Yeah. Ma'am, you're muted. I don't know.
Audio is not working. Video's audio is not working.
They are playing video, but I mean, like, we are unable to hear the sound actually.
Something.
Still not working. The audio is not working.
Hello. We will fix this technical issue in a short while. Meanwhile, we are grateful to have our Director, Institute of Science, Professor A.K.Tripathi, with us today. I request Sir to come and address the assembly.
Good afternoon, everyone. Esteemed Professor Lakhotia, Professor B.N. Singh, Professor Rajiva Raman, Professor Mercy Raman, Mrs. Professor Tikaram Sharma, Dr. Eklavya Sharma, Professor Madan Chaturvedi, and many others I can see on the screen. It's a very special moment to be together on this very special inaugural lecture, which is a tribute to Professor Tikaram Sharma, a teacher who was very, very friendly, as Professor Mercy Raman mentioned. He was very friendly to all, and I happen to have some memories of interacting with him, although not longer duration, but I had a few meetings, and always smiling, as we can see his pictures, always smiling. And to some extent, when I see Professor Eklavya Sharma, I see his smiling face. It's quite similar to each other. So I think it's very nice initiative on the part of the family of cytogenetics and BHU to initiate this kind of series of lectures, and so that we are able to remember such great teachers every year and look at their, the creativity which they have shown in this university and which have laid the foundation of the excellence of Banaras Hindu University. So with these few words, I would like to compliment all the cytogenetics family for initiating this nice lecture series, and I wish that this continues for many, many years. Thank you.
Thank you, sir, for your insightful words. Since we have also gathered here to spread awareness on World Down Syndrome Day and to advocate the rights of trisomy stars and their complete inclusion in the society, I would like to call upon Dr. Tulsi, the founder and director of DEVA International Society for Child Care and today's guest of honor, to share his views and nitty-gritties on the management of the specially abled Down syndrome fellows and its progress over the years and the challenges as they confront us today. I welcome you, sir.
Thank you very much, Professor Parimalda, and all the august gathering of scientists here, and all of them my teachers, my father's friends, colleagues, because my father was professor here in botany department, and my brother was from zoology department, was student of Professor Mercy Raman and Professor Rajiva Raman. And so I'm standing here, I feel quite honored, and also I have a very good memories with Professor Tikaram Sharma, because once he came to know that I am working in the field of Down syndrome and these kind of children, he came to my DEVA center and blessed me, saw all the children. And he said, wonderful work, Tulsi, you're doing. And because we were in the same colony also at one time in BHU, so we knew each other very well. And I used to call him rather more of a Chacha-ji than any other word, because I'm born and brought up here in this BHU. I know each mango tree here. So with this my small introduction about myself, I'm a clinical psychologist and a psychotherapist, deep psychotherapist. And I'm student of one of very renowned professors who is now no more, late Professor Dr. Ratan Singh. And I really owe to him, because he is also one of the colleagues of many friends like Professor Lakhotia and Dr. Rajiva Raman and others. We have very good fond memories of him. So he taught me the various advances after a scientist detects the Down syndrome, the different genetic disorders in a child. Okay, once the detection is done by a scientist, by chromosomal things, genetic thing, markers, next what? Next what? The child is there, the family is there. all the stress is on that family. So when I was in BHU in 1981 to 85, I was doing my psychiatry doctorate in Department of Psychiatry. I was working with Professor Udupa. Professor Udupa told me that you have to go into the community and start doing work in this area because nobody is there. So journey was from 1981. I started seeing Down syndrome children, various disorders, Huntington diseases, children with different syndromes. They're born, detected, now what? Journey led me to one thing, that we scientists, after some time, have to travel 18 inches. What is that 18 inches journey? The 18 inches journey is now what I am traveling. That is from your brain to your heart. That is the journey. Brain we detect, we see things, and then we ultimately come to our heart, what we have to do with them. And then comes the psychotherapy, deep processing of the parents, making them understand, making them clear their misconceptions, making them understand that their child can also become a sort of a nice child, a lovable child. He has no brain, but he has heart. And heart, what you love. So these are the children, Down syndrome children especially. They're the children, they are known as children of God. They are sent for loving. They are sent to be loved. They want your really hug. They always smile. And they can learn very well by a special process of learning, which is known as role modeling. By modeling others, they can learn many things. They can learn even writing, reading, going to shopping, dancing. And today I'm really honored that Professor Parimal said, present them here also. So today we are going to present a small dance recital of these children and also the products which they make it. So in many of the success stories are that these children are earning members of their family. It took long time, say almost to make a child come up to the level of vocational rehabilitation. It takes not less than 20 to 25 years. So the children whom you are seeing now, they are at least 15 years, they are with me. And they are not like this. They're just babies when they were brought to me. So with this, thank you once again for giving me this chance. And I'm really very happy. And I am also, भाभी जी पता नहीं मुझे पहचान पाएंगे कि नहीं, एकलव्य मुझे पहचान पाएंगे कि नहीं, because I know Eklavya very well. And Eklavya, I think he was in my school. And
I remember you very well.
थैंक यू सो मच. मेरा प्रणाम आपकी माता जी को भी और सभी को. और ये अभी प्रोफेसर मर्सी रमन ने पूरी मेमोरीज एकदम हम लोगों की ताजा कर दी, पूरी मेमोरी लेन में ले जाके, और आपको देख के तो और ताजा हो गई.
Dr. Tulsi, can you hear me? Your Bhabiji is here.
हाँ, so nice. थैंक यू सो मच. बहुत अच्छा लगा आप लोगों को देख के और मिल के. इट इज रियली ए प्रिविलेज, ऑनर. थैंक यू सो मच एवरीबॉडी.
Sir, can you explain more on role modeling topic that you told?
See, in training of these children, the main therapy which we use is known as behavior therapy, altering the behavior of the child. There are two scientists who have worked on behaviorism. One, all of you know, he was a scientist, Pavlov, classical conditioning and instrumental learning models, and also Bandura, who is worked with Montessori, Montessori all of us know about school education. So Bandura was the proponent of role modeling. In role modeling, we all copy. All of you copy someone or others. You copy your mommy, you copy your papa, you copy your hero. So we copy somebody. So with hairstyle, standing, even I can tell that seeing a person that yes, your papa listens to you like this because you have copied him. So in this way, these children also, by gestures, by stimulating them and reinforcing them with positive rewards, we make them into social skills, namaste, talking, speech, and everything. They have some problems with speech because of their tongue and the tongue structure of the tongue, but still by certain sounds, certain speech stimulation methods, we can train them in minimal speech, expressive speech skills. And also they can so if I am able to answer this, that changing what behavior therapy says, changing the negative behavior into positive behavior. How do you change? We call it slicing method. If a behavior has to be changed, we slice it into smaller units, and then each unit we start changing it, and then shaping it. So in this way, slicing, shaping, the behavior from a negative side, child who is not able to sit down, child who is not able to pay attention, concentration, child who is not able to talk or hold anything, we can bring him to the children who are now you are going to present it here, who will dance on the sounds by seeing their model. There is a model with them who will be there, their teacher, and that teacher will little bit signals, and they will dance, and they will enjoy. That's it. Thank you so much.
Thank you, sir, for highlighting the importance of collective understanding and global conversation about Down syndrome, which will empower people around us to advocate for full inclusion in society for people with Down syndrome and other disabilities. I request Professor Parimal Das, Coordinator, to kindly felicitate Dr. Tulsi. Thank you, sir. I think the technical glitch has been fixed, so can we play the video, Professor Sharma's video?
I, Mrs. Sharma, wife of Professor T. Sharma, got a call from Professor Parimal Das and his wife, Dr. Bhagyalakshmi. They said their Center for Genetic Disorders is organizing first Professor Tikaram Sharma Memorial Lecture and World Down Syndrome Day on his birthday, 29th March. They invited me and my family to join this program online. They wanted us to say something about him. What should I say when these days in every news channel I hear the word "cube" most of the time? This word I had heard for the first time when Professor T. Sharma was going to Kiev to attend International Symposium on medical aspect of Chernobyl accident in 1988 as an expert representing India. So these days, Professor Sharma is very much alive in my memory. How he described that nuclear disaster will badly affect the health of the people generation after generation. He always described us about the places he visited when he went to different places to participate in international seminars, symposiums, conferences, and meetings. He was open to all, free, frank, and jovial, but sometimes it was quite difficult for all of us to face him. Professor Sharma always wanted guests or his friends to welcome them at home. Professor Kasperson, Professor Onu, Professor Sperling, Professor Hansman, Professor Onu, Professor Ken Jones, and many others had lunch or dinner at our BHU quarters. He had no hesitation inviting them, whether they will feel uncomfortable in Indian homes. Today, when he is no more around us, his distinguished students carrying his legacy further. Professor Rajiva Raman, his first PhD student and later on his colleague, Professor Mercy Jacob Raman, his student and colleague. We all remember you both and your family and the days we spent together in Banaras. Dr. Tulsi ji, you must be fine with your family. Once you had invited Professor T. Sharma and me at Diamond Hotel, where lovely kids of your NGO were performing beautifully. Your dedication and utmost care has given them freedom and exposure to the outer world. It is so heartwarming, so praiseworthy. Can it be fully expressed in words? On this very special day, I want to thank Professor Parimal Das, coordinator of this program, and the whole committee members for inviting us to be a part of this important lecture. I thank you all and wish you all very healthy and happy life.
Sometimes I look up in the sky and say, we love you, Baba, and miss you always. Thank you for touching so many lives. We love you, Baba.
I'm Shania, Professor Sharma's granddaughter. And I'm Raghavir, his grandson. Baba has always inspired me to never be afraid in doing anything, whether it was something as simple as helping me pull out my first baby tooth or showering me with words of wisdom before my black belt exam. Baba has always encouraged me to view my fears in a different way, in a happier manner, which I will always remember and cherish forever. Baba has taught me that no matter how many struggles and hardships we go through in life, we can always follow our dreams and do what we love.
I'm Sushant, Professor Sharma's younger son. The void that he has left in me can never be filled. I wish only if he could still be around. He was a very caring father. I miss him a lot.
My father, he was the most loving, caring, and a very encouraging person. In fact, he was the pillar of strength for all of us. In joyous moments, in difficult times, we always miss him. We know that his blessings are there for us forever, but still we miss his physical presence.
I'm Subhashish, Professor Sharma's elder son.
I'm Rashna, his daughter-in-law.
The one thing that really inspires me about Baba is his courage. As we were growing up in Banaras, as young kids going to school, he was always our pillar of strength, and we looked up to him. No matter what the obstacles, no matter what the challenges, He was always courageous. He always spoke his mind. He never cared about what others would think of what he said. We really love to follow his footsteps.
I have cherished every moment I spent under Baba's guidance. He was and he is my guide and my guru. Baba always believed in what I did, not just as a person, but also as a practitioner. We as a family are definitely very fortunate to have received his blessings, and we continue to feel and perceive his divine presence.
On behalf of the entire Sharma family in Kurseong, in Siliguri, and here in California, we want to thank everyone in Banaras Hindu University organizing this memorial lecture in honor of Baba. We miss him and wish you all the best for your lecture. Namaste.
Also, his colleague, Professor Karl Sperling, have sent us a write-up about his individual experience with the professor. So, we'll like to play that. Professor Karl says he met Professor Sharma first time in Berlin, and that meeting had led a lifetime of scientific and personal relationship between the two. So let's just see what is. I can read one? Okay. Okay. They can see the screen, ma'am. Okay. From first I should read? Okay. Dear Professor Das, dear colleagues and friends, warm greetings from Berlin to Varanasi. On the occasion of Professor Tika Sharma's 87th birthday, you present this first memorial lecture. What a wonderful idea. Here are a few personal reminiscences of this distinguished scientist, dedicated university teacher and educated, warm-hearted person signed off by Karl Sperling, Institute of Medical and Human Genetics, Berlin. We first met each other in the early 70s on his visit to Berlin. Since then, we stayed in close contact. We were both cytogenetics, working amongst others on rodent models with respect to heterochromatin and on radiation-induced mutations. Tika Sharma provided us with cells from the Indian manjak, which has the lowest chromosome number of mammals, with only 6 chromosomes in the female karyotype. This resulted in a series of publications. Prematurely condensed chromosomes of the Indian manjak of the G1 phase of the cell cycle. As you can see in the picture, the interphase chromosomes of the manjak were obtained after fusion between human mitotic cells and manjak G1 phase cells. This figure was the cover of the journal Biology in Unsere Zeit 10, 1980, and reproduced in the textbooks of Kling and Cell Biology 1984, Trott Chromosomen 1991, Rao Johnson Sperling, Premature Chromosome Condensation, Application to Basic Clinical and Mutation Research, Academic Press, New York 1982. One special gift of Tika Sharma to our institute was the mediation of Rajiva Raman in 1980 as Fellow of the Alexander von Humboldt Foundation. Here Raman sir is there on a boat trip with members of the institute participants. One special gift of Tika Sharma to our institute was the mediation of Rajiva Raman in 1980 as a fellow of the Alexander von Humboldt Foundation. Another gift was the mediation by Rajiva Raman, office student or Tika's scientific grandson, to our institute, Indrajit Nanda. He highlighted one point. A highlight of this meeting was a boat trip on the Ganges early in the morning. I filled an Indian water carrier pot with holy water from the Ganges, which has a prominent place in my office, in my home office, and always reminds me of India and Tika Sharma, Rajiva, and Mercy Raman. Tika and I both are convinced that cytogenetics is not a purely descriptive science, but that it can explain fundamental relationships at a higher level of biological organization. This also makes the difference between seeing and watching. Watching requires a theoretical understanding and therefore goes much deeper. In the Song of the Lincius, A Watchman Faust II, 1832, by Johann Wolfgang von Goethe, this difference between seeing and watching is aptly expressed. The watchman speaks this poem in retrospect at the end of his life. I think Tika Sharma would have wholeheartedly agreed with him. We also have one of Professor Sharma's student, Professor Cheong Nge, who has also sent us a write-up. Please have a look. Professor Sharma accepted me as one of his PhD students in 1980. Since then, I was introduced to the family of cytomets. Over the years, we had learned from Sir not only the required scientific knowledge, but also how to behave and be responsible as a member in the modern society. I thank them. Now I request Dr. Bibha Mishra to come and share her thoughts.
Years from now. I was adopted from obstetrics and gynae exactly 50 years from now. I remember my first meeting with Dr. Sharma. I pillion rode with late Dr. Manjeet Singh. I think Rajiv remembers her. In the 6 o'clock in the evening, and I was introduced to this short man who was sitting, got up from the chair. Hello, doctor. That man who's calling me, hello, doctor. I'm still an MD student. And since then, that association of that long 50 years continued till he was in Varanasi. Sometimes, after 1.5 years, when I submitted my thesis in cytogenetics in repeated abortion, I had a disaster. I came to him running. Why do you worry? I said, why not worry? I am failing in the examination without appearing. There should be a way out. Let me see, what can I do? I said, I don't have time. I have exam 7 days ahead. And that too MD, obstetrics and gynae, is such a disaster exam. Out of 16, only 2 pass, 14 fail. And I am failing without appearing. He said, don't worry, you will pass. No, how can I pass? You do one thing, come with the copy to me. I reached 9 o'clock to him in this lab only. He did not do anything. Rajiv was there working, and Mercy was sitting in one corner behind this thing. Come to my side. I showed him this thing. What is the query? No query. Your thesis has been rejected. Why? Give me the point. I gave him the point. Don't do anything. Take out these 10 pages from this your thesis and 10 more pages to your thesis. Then how will I get your sign because you are leaving? Don't worry. You keep the first page and add it to the rest of the page. And that day changed my life. I did the same thing what he did, said, and resubmitted the thesis and got it passed. And I appeared. Out of 16, I was the only one to pass. Had I failed, the life might have been different. And the things did not end there only. Many times I'll find him in my chamber. When I'm seeing the patient, suddenly Vibha, Dr. Vibha, and I'll see Tikaram smiling at me. The association went on that long, and I can never forget. आज की तारीख में मेरा उनको अभिनंदन प्रणाम जो कुछ कर सकते हैं करेंगे. I am just here because of him. Thank you all so much.
Thank you, ma'am, for sharing your memories with us. Now we'll have the performance by Trisomy Wonder Kids from DEVA International Society for Childcare. I request them to come forward. इतनी सी हंसी इतनी खुशी इतना सा चांद का चल बनाए आशियाँ इतनी सी हंसी इतनी खुशी इतना सा चांद का ख्वाब में चल बनाए आशियाँ हरे 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 जिंदगी हरे लगा के चल सुंदरा आधी आधी बात आजा दिल की रेजा थोड़ा सा तुम्हारा मेरा भी होगा अपना ये आशियाँ किसी किसी खुशी चांद चल बनाए आशियाँ
किसी हंसी किसी खुशी कितना सा टुकड़ा चांद का से चल बनाए आशिया किसी हंसी खुशी सा टुकड़ा चांद हाँ वो उनको चल बनाए आशिया.
Thank you for a beautiful and heart-touching performance by the very talented kids. I would request our coordinator, Professor Parin. Thank you for a very beautiful and heart-touching performance by the very talented kids. I would request Raman sir, please felicitate the kids. Today we have among us our keynote speaker, who is the first Professor Sharma student, Professor Rajiva Raman, Distinguished Professor, Department of Zoology, BHU. Owing to his prominence and being an excellent orator, the lecture series has attracted many attendees across the globe to this event. Sir will give us a keynote lecture on animal cytogenetics in India in global context. I request Sir to please take the dais.
Oh, okay. Just right. Or other like this, right? Tick or right. Uh, Good afternoon, friends. Professor Tripathi, the Director of Science, Professor Lakhotia, Professor B.K.Thelma, Professor Singh, Professor Mercy Raman, and a band of young colleagues, very young, plus the wonderful audience we have online, which includes Mr. Sharma, both and we have got Cheong, Bhudev, and so many Madan, and many more. All of them who are here, we greatly welcome you to this evening. In fact, after such a wonderful performance of these special Downs children, having another lecture actually loses a little bit of its importance, because we have had the best of it. Nevertheless, I feel it's an opportunity that I should grab with both my hands to be asked to give the first Professor Tikaram Sharma Memorial Lecture. It has been, this credit comes to me because I happen to be his first student. And I feel I thank my stars, because I could have missed this opportunity by whisker. When I came to join PhD with him, I was asked to make grasshopper meiotic chromosomes by air drying preparation. We used to make squash preparations, as they were called, and it was very easy. But when asked to make air dry preparations, a single testis having to centrifuge 3-4 times using Ford and Evans's technique. It took me about 3 months and I was still not getting good preparations. So one day I went to him and I said, rather sheepishly, I'm your first student and I haven't done anything good for these days. If you like, I can leave. He said, how much time have you spent? And I said, it's going to be 3 months. His reply was characteristically his, which I followed on later on. He said, oh, I couldn't do anything for 6 months. Protein polymorphism. In 1972, it was still a new thing. And I thought if I can mix both the things, chromosomal as well as protein polymorphism, it will give me a good idea about how evolution proceeds. I went to Delhi University in the Department of Anthropology, learned a few techniques, how to do electrophoresis through different char gel, cellulose and state gel, polyacrylamide and everything else, and came back very enthusiastic. And when he came back from summer vacation, I told him that I want now to proceed further with protein polymorphism. He said, no, this problem was mine. I gave you, as a PhD student, a PhD problem. I will pursue it. You are qualified to do your research. Now go and search a problem for yourself. I was very disappointed with that teacher. Having respected him for so long, have done so much, now he says, go and find something else. He added, he said, if you continue to work on rat and this and that, you will always remain somebody's student. You will always remain just extending your PhD problem. If you want to be in science, better find a problem for yourself. I think that was the best advice I could ever get from anybody for my career. That was Tikaram Sharma, a teacher par excellence. Everybody knows that. But he was a die-hard scientist. Chromosomes of Munjak, which you saw in Karl Sperling's slide also, were first reported from America by Kurt Benishke in 1969. And Professor Rai Chaudhary, rather jokingly, I believe, told Professor Sharma, who was then associate professor, reader, said, Tikaram, this Munjak comes from Darjeeling and the chromosomes come from California. What does he speak about us? Next Durga Puja vacation, he goes back, goes hunting, hunts a buck, it's a male, puts him on his shoulders, runs 5 kilometers or so, comes home, takes out the testis, doesn't have a fixative, takes out a bit of vinegar, takes out a bit of brandy, which a Nepali family would not normally have, makes a fixative, fixes the tissue there, cancels his visit, comes back to Banaras, makes the chromosome preparations. And I remember that day very well because I and Lalji Singh were doing in dark room, we were making automatic film application. And somebody was thumping at the door of the dark room and we were annoyed. He said, wait, but that fellow doesn't wait. And he opened the door and found Dr. Sharma standing and he said, chromosome dekho munjak ke? That tissue which he had fixed there, he had made chromosomes from. This slide will of course show you the first 3 students of Professor Sharma. Myself, on the other side of it is G.S. Garg, who was the second student. And on my side is Inder Gadi. Sadly, Gopal Garg is no more with us. Dr. Gadi is in US, in North Carolina. He's one of the most established clinical cytogeneticists that USA has. And then, of course, the next slide I'll show you. This is the photo of that day when do you have the pointer, please? Can I use? Okay. So this is the spermatogonial. This is the spermatogonial. This is the spermatogonial plate from that testis. You can see metaphase 1 here. You can see the trivalent below at the one which is XY and an autosome. This is the interesting chromosome that they have. This is the lowest number of chromosomes that any mammal has, and this is the anaphase 1. So typical meiotic preparation. This was the first time it proved that what you are seeing as somatic chromosomes were actually what was also seen in the meiotic preparations. That was his science. But as Mercy was telling in her presentation, he was a man of as an institution builder. So a couple of these slides. This is a slide from the same conference of DNA repair meeting that Mercy was talking about. You can see in the front is G.H. Taylor. I don't know how many of you are familiar. The one person who showed for the first time semi-conservative replication. I'll come back to that again. Much before Westerton and Stahl did it. He did it on chromosomes. And when what is his name, Delbrook, he was in Japanese genetics conference. He heard Taylor presenting his work. He came back to California and locked Messelson and Stahl in the room and said, unless you complete your manuscript, and I sent it to PNAS along with this manuscript, you will not come out of that room, because they had done the DNA experiment on semi-conservative replication. That is Taylor, and of course you can see Professor Sharma. You can see Thelma here. Some of you who have heard the speaker are speaking, and you can see Dr. B.C.Das. This is another workshop. I was then out in Germany as a Vornenbold fellow, and I went to cell biology meeting, and unfortunately, I found even I went to a chromosome conference, and I found nobody was talking of chromosomes, and they were all talking of molecules, DNA, southern RNA, and all that. And I felt frustrated, and I wrote to a colleague of mine that later on became my wife, that I'm feeling very frustrated. What life is this? What kind of science am I doing if I'm not competing with all others in the job? She took that information to Dr. Sharma, and he said, before he comes back, you should have molecular biology lab ready. Of course, it took a little more longer, but he did arrange a workshop. You can see these people there. I'm sure you can identify Subhash Lakhotia there. You can see Lalji Singh there. In the first projection, you can see Ken Jones, Thelma, Bhudev. They were all participants. Myself as a participant, Howard Cook. And then below, you see another of the slide, which was how to do PCR, which is called as workshop for microcloning. And you can see your own coordinator standing there as one of the participants. He's diminutive, so I don't know whether it's easy to recognize him, but I think he's somewhere here. So you can see him. So that is what was organizing institutions. One person who made a difference to his life and to the life of all of us was the one sitting by his side. That is Professor S.P.R. Chaudhary. He was the one who brought actually animal cytogenetics to India, and that's why I thought it will be in context if we see this slide and also the next one. This is a rather busy, I mean, an old-looking slide. Go back. I can say. So, will you mute yourself, friends, on the online? Yes. I'm sure you can't read much from it, but it will give you some idea how the tree flourishes. The phylogeny of cytogenetics lab. You can see at the bottom here, you can see S.P.R. Chaudhary at two places, here and here. This is S.P. Rai Chaudhary in Calcutta. This is S.P. Rai Chaudhary in Banaras. And in S.P. Rai Chaudhary in Calcutta, you'll find up here the name A.S. Mukherjee. Even if you can't read, please follow my arrow. And a little above, you'll find S.C. Lakhotia. Mukherjee did not complete his PhD with Rai Chaudhary and he went abroad to Kurdistan. But one of his students was Subhash Lakhotia, who happens to be with us here now. And you see a very big tree coming from him. Somewhere you see J.K. Roy in between is here. Similarly, if you look at this tree, you'll find T. Sharma here. Then you'll find Professor J.P. Gupta, B.N. Singh, Mercy Raman. From T. Sharma's stock, you find Rajiva Raman here. And then you find from Rajiva Raman's stock again, a few names. This is an old slide. You can see the old paper. It was of year 2000 when Dr. Sharma became 65 years and we had a small function in the department. It gives you the sense of what is called as cytoghrana. That's what Dr. Sharma used to call it. And this ghrana has become a mahal, or whatever you call it. It expands and expands, and it is all credit to you all that it expands. I will now come to the topic, which is animal cytology, cytogenetics in India. What I want you to understand is that the two persons who made a fundamental difference to the understanding of modern biology were the people who are asking the question, why are we similar and why are we different? We are similar, but never the same. And yet, we are similar. The two people who are asking this question, one of them was Charles Darwin, see, who found or convinced himself and others that the variability that you see around is not created, but is a continuum of a process following, and that process is called organic evolution. There was another person, less recognized and died unsung, was a monk in a church in Prague called Gregor Mendel, who wanted to be a teacher of mathematics, could not pass out the exams, and therefore took up the job of a teacher in a parish, but did some experiments on pea plants, and he said that there could be reason for this variability, and there should be a reason for our similarity. And he said that there are factors which take care of it, and they work. But today's talk, I will not touch genetics. I will try to confine myself to cytogenetics and to tell you as to how genetics got integrated into cytogenetics. But at the time when Mendel or Darwin were working, there were also people who were working on nucleus of cells, and they could show certain of the features. As you would see here, this is called as the first picture of what we call now as polytene chromosomes, was drawn by Balbiani in 1881. He did not know definitely that they are chromosomes. He did not know what chromosomes are, but he drew this picture, which later on was proven by that it was from carinomus salivary glands, and that they were chromosomes. The next very important finding of that period would be that of Fleming of 1882, where he drew with absolute precision how mitosis happens. Understand, there were no good microscopes at that time. There were no cameras attached to the microscopes, and he drew them using his methods, as we'll see. He used the word mitosin, which was later on called as mitosis. And also, the threads that they were seeing, because he was able to stain them, he called them chromatin, which was later by Bahadur was called as chromosomes. So what we know today as chromosomes in 1888 was actually shown very beautifully by many, but most clearly by Fleming. Also, in around the same time, working on oocytes, there were two people, Fleming, who was using salamander egg and showed large chromosomes, that he called as giant chromosomes. He didn't know there were chromosomes again. And then there was another person who worked on fish, Ruckert, in 1892, who called them, because the shape of the appearance of the object that they were getting was like a lamp brush, so he called them as lamp brushes. And then the X chromosome was also discovered before the last century, and that was by Henking, who was working on a bug. And he was working on the meiosis, and he found that whereas other chromosomes were looking like paired, there was at least one which does not have a pair. The poor fellow was low and alone, and often when you don't find a good name, you begin to call things as X. And so the name was given as X chromosome. It was seen in male testes. This is a picture, original picture from Walter Fleming's 1882 paper, and you can see those of us who do mitosis, how beautifully, this is a camera lucida drawing, I believe, and everything is so nicely done. So, so you can see this. I guess better keep it full because I don't want it to be, how will it become full? Yeah, leave it like this, no problem. But as we know, since most of us are here students of genetics, that the laws of Mendel remain unknown until 1900, when 3 independent scientists found out nearly similar observations on different model systems. And this found that what was that therefore it was recognized as laws of inheritance by Mendel. But where are these factors present? And in 1902, there were 2 papers. Actually, Sutton's paper came out in 1903, and Bowery's earlier. We give a lot of credit to Sutton, and we forget about Theodore Bowery. But Theodore Bowery had worked for 10 full years and shown how the chromosomes go from generations to generations. And even if because he was working on sea urchin chromosomes, it was easy to modulate them. He could remove a chromosome and he could show that they could lead to abnormality. One. Secondly, he could show that it is still inherited. Secondly, then of course as we see, and it's very nice to read them. Bovary said a specific assortment of chromosomes is responsible for normal development, and this can mean only that the individual chromosomes possess different qualities, because different chromosomes are different. And Sutton said the association of paternal and maternal chromosomes in pairs and their subsequent separation during the reduction division may constitute the physical basis of the Mendelian law of heredity. While working on insects, they also found, in addition to the X chromosome, which they had seen, which was seen by Henke, there was also another chromosome which sometimes did not pair, occurred only, it made a heterobivalent in the male meiosis, but not, there was no such chromosome in female. And that's why an additional name came from Wilson's. Wilson was a mentor, teacher of Sutton, also a medical doctor, and as you would know, Wilson and Morgan were in the same institute and they were very good friends. And that's how when Morgan came, so there was a big good team of people in Columbia at that time that started genetics. Morgan genetics, Wilson with most of the chromosomes. So that's how the chromosome Y came. And then of course, what Mendel did not show, or perhaps purposefully ignored, was linkage and crossing over. This in spite of the fact that he had some crosses which did not explain his laws as well as he wanted to. So this concept also came, and as we'll see later on, how chromosome work. And to understand this point very clearly, there were no good techniques available to make chromosomes at that time. Histology was the only way. So the most of the knowledge that you have of chromosomes, at least until 1923 or 1930, are from the sections, and the sections have been redrawn. It has led to errors, and it has yet led to very good findings. Now, if you look at that, you find this period of 1911 to 1950, from the chromosome point of view, was a period which set many, many thoughts. First of all, it showed clearly that the sex is determined, at least in Drosophila, only chromosomal. So chromosomal basis of sex determination came out from Bridges' work in showed the importance of chromosomes. But even more importantly, I rate it very, very high. Bovary, through 3 very classic papers in 1913 and 14, working on sea urchin chromosomes, made a prophetic, prescient prediction that malignancy is caused by chromosomal rearrangements. As you can understand, he had not seen human chromosomes. He had no idea. He had only sea urchin chromosomes. But he knew that malignancy occurs. And he said at the basis of the malignant tumors, there must be chromosomal rearrangement. As you know, Bovary died very young. In 1915, he already died. But by that time, he had made landmark contributions to chromosome biology. Then in 1928, Metz published a couple of papers on Sierra showing how chromosomes can get eliminated in a regulated manner. Only one parent's chromosomes can get eliminated, and others will remain intact. And that's what was shown later on, called as chromosomal imprinting by Krauss in 1960. These are, in essentiality, these are chromosomal phenomena, which have had a genetic explanation later on. The polytene chromosomes, the chromosomes that were shown by Balbiani in 1882, for very long, it was not possible to actually find out as to what they are, until later in 1930s, that people from two groups of people, Bauer and Heitz in 1933, drew the chromosomes and said that they were indeed chromosomes. They are part of the nucleus and they are chromosomes. Okay, I must mention that Balbiani, I think I wrote it here, called that structure that they showed as permanent spirine, which was later on shown to be chromosomes. Now, around the time when Heitz and Bauer showed in 1933 Drosophila chromosomes, Drosophila melanogaster chromosome, polytene chromosomes were drawn by Painter again the same year. Now, it's interesting that they still did not call it as, they called it as giant chromosomes. It was in 1935 that P.C. Kohler in his paper wrote that they are showing multiple threads and therefore they should be called as polytene chromosomes. But in the same paper, he's so nervous that he said, I will not call it as polytene chromosomes. I will still call it as multiple threads, but a good name would be polytene chromosomes. In 1935, Bridges actually showed that you can map genes on chromosomes. So what you were seeing as band or patterns that you were seeing in Drosophila or polytene chromosomes were actually reflecting genes. It was also shown by Muller and others that there could be the chromosomes can break, they can show non-disjunction, so the chromosome numbers can vary, chromosome structures can change, so they can become translocation, there could be inversions and all, and these changes could have an effect on the phenotype. Well, yes, one important finding was again in 1923 by this another painter, not the one that we are talking about in Drosophila, by Theophilus Painter, again testis chromosomes, and he identified as 2 and 48. Two independent people, P.C. Kohler also in Edinburgh, showed these and counted as 48 chromosomes. Now, there has been a lot of debate on this as to why did they commit that mistake. Now, don't forget, they were still cutting sections and they were trying to put the sections together and in drawing them. What people suspect is that whereas P.C. Kohler had an individual where I think X and Y had gone separately, and that's why he counted them as 24, whereas Painter, while drawing sections, that's what Frank Ruddle found out later on, perhaps saw chromosome number 1, which is so large, as 2 different chromosomes, and that's how he counted as 48. The reason that I gave this explanation to you is because, after all, a lot of people make fun of human geneticists. You couldn't even count 46, they say. So, and then McClintock talks of jumping genes. Don't forget, the major change in the technology of chromosome preparation came in 1925 when McClintock did squash preparation. Until that time, there were only section cutting. So squash preparation, which we do routinely in our laboratories, was the one developed by McClintock in around 1925. And while she was a master person, she was not an animal cytogeneticist, but still I mentioned this because she gave a very big concept, that of jumping genes. We all know, even now we find it difficult to believe that something as static as chromosome could have mobility. She talked about, because she showed how certain points in a chromosome, they were heterochromatic blobs as we used to call them, could move from one chromosome to the other. And don't forget, this hypothesis she gave after seeing the maize plants. So after seeing the phenotype, she thought that there must be a chromosomal movement. Then she prepared chromosomes and showed that yes, there are parts of chromosomes which keep moving. Later on, of course, she got Nobel Prize 40 years later. Sometimes there's a virtue in becoming old. And then, of course, in 1949, Barr and Bertram showed they were working on neurons of cat for finding whether there are different kinds of neurons. But one thing that they found, which was very important, was that the female nucleus could be distinguished from the male nucleus. So you could distinguish sex just by seeing the nucleus of a cell. That the nucleus of a female had a chromatin body which the male sex lacked. And therefore you could do nuclear sexing, which was later on called as Barr body. But so I want to show you this picture drawn by painter again, by camera lucida drawing the Drosophila chromosomes. You can see how precisely, how beautiful job has been done. You can also see in chromosomal rearrangements drawn again here. You can see here, these are chromosomal rearrangements. And look at what the editor wrote on this paper. Ever since the gene hypothesis was generally accepted, geneticists and cytologists have dreamed of the day when it would be possible to see actual genes instead of having to be satisfied with studying their shadows, which were reflected in the morphological development of generations of organisms. Giant salivary gland chromosomes of the fruit fly are proving to be a new genetic tool of the utmost importance. That indeed it was. This is the physical map made by Calvin Bridges. Bridges improved upon this map several times. By 1942, before he died, the map that he made has actually very little has been improved upon that map with Bridges' map. And there he could show what you see as bands were actually reflecting genes. So that was the great contribution of Bridges, and how the chromosomes, these two slides that I've just shown to you, I borrowed from Professor Lakhotia. But I still feel the decade of 1950s was the watershed decade. We have always talked about the first half of the 20th century as the half that belonged to physics, quantum mechanics, relativity, atomic bomb, and what have you. This decade of the post-war decade is an important decade from our point of view, as you would see that in 1952, Behrman with Bauer and also Pavan from South America, they showed that these polyethylene chromosomes have got certain regions swollen, which are not permanently swollen, but they get swelled up at some stage of development, and later on they don't do. And they suggested that these swollen up regions, which they called as puffs, should be sites of active function. So when the gene function occurs, you can see under the microscope as the chromosomes, as that region will swell up. I'll show you a slide immediately after this to give an idea of that. The other point that I discussed, I showed you the picture of J.H. Taylor. J.H. Taylor published it in 1957 in the PNAS. This paper was published. A companion paper was that of the Merselson and Stahl, which you all study in our class books as the evidence of semi-conservative mode of replication. But using labeled thymidine and allowing the chromosomes to Viciafava chromosomes to grow for 2 cell cycles, he could show quite clearly how a chromosome labeled, if you allow it to grow for 1 more cycle in absence of thymidine, the label will concentrate on one of the 2 chromatids. And if you go another cycle, you'll find that this will become 1/4. Using this piece of evidence, he showed that the replication is semi-conservative. He also showed that possibly only a single DNA molecule runs through the length of a chromatid. Now, this was a debatable issue, because people were at that time wondering whether chromosome, which is so thick, whether it has got a single DNA molecule, having accepted DNA as a genetic material with great difficulty. People were also saying, oh, there can't be such a thick chromosome, it can't be just some single molecule. And therefore, there was a debate whether it is polynemic or unanemic, whether it has more than 1 threads or it has got 1 thread. Taylor, in this paper, very beautifully demonstrated that there is only one DNA molecule because of the semi-conservative mode of replication. But he was so overtaken by the prevailing evidence coming from electron microscopy and so on, from the work of Hans Reis and others, that he said, I am not saying that it is unilinear. And he tried so many explanations to say as to what he is showing could be actually polynomial, even though he had shown it to be unilinear. It's an interesting paper to read and a wonderful experience, how in science sometimes the dogmas become so overbearing. One of the dogmas was, as you would know, that human chromosomes are 48. Until 1956, when Seo and Levin published a paper showing that human chromosomes are actually 46. The interesting part of it is that Levin, who was a cancer cytologist, had a few technicians with him, and he gave them a couple of slides to observe and say how many chromosomes they have. And the girls, as they were young, they said, we are not able to count 48. We are counting 46 chromosomes. And the fate was that he made those girls to run away. He said, you can't even count 48. What research you will do? Then he had an MD doctor, Seo, and Seo stuck to his guns and he said, no, this is 46, not more. Then Levin sent this picture and told other people like T.C. Shu and others, we are getting 46. What are you getting? Interesting story is that Shu had a picture on his wall, paint frame. He went and counted and he found it was 46. He had written human chromosomes 2 and 48. Actually, there were 46 chromosomes. That's what dogma does. So that is what Seo and Levin for once settled. Now look at the importance of this finding. Within 3 years, in 1959, as you would see, 3 different chromosomal syndromes were found because they were syndromes where the chromosome number had gone beyond or less 46. A chromosome more could lead to Down syndrome, Klinefelter. Down syndrome was shown by Tulsi in France, whereas one chromosome less than 46, 45 led to Turner syndrome, which was shown by Patricia Jacobs, and John Hamilton showed Klinefelter having a chromosome more, but not the same chromosome, which led to Down syndrome. And that's why both the chromosomes were pretty tiny. The chromosome, I mean, the one that got Downs had a tiny little chromosome, but the one that was missing was a large chromosome, the X chromosome, and where the Klinefelter had. So it became important that we should have a standard karyotype so that internationally people would know when they are looking at one chromosome, anywhere else in the world, wherever you look, you know that which chromosome is being talked about. And therefore, the Denver conference was held in 1959. It was published as the proceedings were published, where the standard human karyotype was accepted, and that's why it is called as Denver Conference karyotype, which was of course worked upon much later, and so on. Now, in all this, what I'm trying to show to you is, okay. So now this is from the, you can see this is the DNA, the Mona Lisa of modern biology. You can see them how they look like, and I think all of us would agree there isn't a more beautiful molecule than the DNA, right? So that's why it's very apt to call it as Mona Lisa. This is a slide again from, I'll come back to this slide again, but this slide I'm showing you at the moment only to show you, for example, if you look at this portion, this is not swollen, but it gets swollen here. It's much better here. You don't see the symbol thread, but the same portion gets swollen here. And it's swollen because it is incorporating a lot of uridine, tritium labeled uridine, which means that it is transcriptionally active, which means that this swollen portion, what we call as puffs, are indeed functioning genes, which was shown here. Well, this picture is of not of 1960, not of between 1951 and 60, but it's a picture of much later. But as I had shown you in the previous slide, that Lambrusch chromosomes were chromosomes 1, and that they were the active state of chromatin was already demonstrated by Joseph Gall and Callan from England. But I think the most elegant pictures of this came from Oscar Miller in 1966, 67, and then of course a number of pictures that he brought out. This is a picture, as you can guess, from the oocyte of an amphibian. And as you know, amphibians have got a lot of yolk and they have got a lot of RNA made. So this is, and for protein to be made, you need a lot of ribosomal RNA. So these are these amplified figures of amplified RNA of 45 RNA, and you will say, you could see very easily that there is a Christmas tree and then there's a small gap and then the Christmas tree. One such Christmas tree is actually 145S rRNA gene, which is, and you can see at the axis, you can see RNA polymerase sitting here, and from this slow to this small to this big, you know, this is RNA which is being transcribed on this template, and that's why you can't have a better demonstration of a gene in function than this historically beautiful picture. Now, all this while, yeah, this is of course the same famous picture of Theo and Levan, which was published in Heritage, the journal which doesn't exist anymore, where they could show 46 chromosomes. This is a picture from Jacob et al., Patricia Jacobs' paper of Turner syndrome, which showed that there are 45 chromosomes. You can see a single X chromosome. She was not very clear whether it is X or not. Therefore, there's a question mark. And then, of course, this is a picture of a Down syndrome, which is from the work here. Most of the pictures henceforth that we will show, most of them would, I believe, will be from our labs, the work done by colleagues in CGD or in zoology or then somewhere. So that is a period where, I call it the watershed period, because I realized that that was the time which set cytogenetics, especially in the clinical cytogenetics, its path. So clinical cytogenetics started from 1951 onwards, and therefore, now I also wish to put to you two very papers that I like very much. One, of course, that of Watson and Crick showing DNA structure. I show this slide to you for one, that you can be very short and you can still make the biggest discovery. One paper, one page paper, you can see. The other paper that I put to you here is that which propounded what is called as Lyon's hypothesis, where she showed using very simple methods that one of the X chromosomes in mammals gets inactivated. Don't forget, they never talked about dosage compensation. Perhaps they did not even think of dosage compensation at that point of time. It was earlier shown in early 30s in Drosophila. So this part of the talk I wanted to devote to what has been known until 50s, and then came, and I will take very little time on this, a remarkable change in chromosome biology, where a simple technique by treating the metaphase chromosomes with trypsin could lead to a pattern on chromosomes which will be banded and which will be chromosome specific, which is important to understand in this, that this was not true only for humans, but this was true for every other species that you can do. So they were permanent structures that could be developed when you give a certain kind of treatment, hot water, trypsin or whatever else. Of course, that's also spoke about the chromosome structure, of which I'm not going to talk in this talk, because this talk I thought I would confine only to the historical aspect and the lighter aspect of chromosome biology. This banding, as you can see, this is, you can, well, not only that you can do trypsin, but you can also use certain fluorochromes, which can be used, and they would also behave like banded chromosomes, as you can see here in this case. This is a chromosome, the first chromosome that you see here, or you can see here, this portion is showing staining and not anything else. That is heterochromatin, cytochromate heterochromatin. This is what we call as trypsin banding, what's called as Giemsa banding. In this case, you have treated the chromosomes with a fluorochrome coil. It's a benzimidazole derivative, which is looking exactly like the G banding. And then there would be another drug like olivomycin and others, antibiotics, which you treat, which will show a reverse banding of what you're seeing as G banding in this case. So the whole chromosome organization can be understood through differential banding patterns. And then a lot of newer technologies have since developed: fluorescence in situ hybridization, chromosome painting, chromosome dissection, comparative genomic hybridization, spectral karyotyping, CGH microarray, and so on and so forth, which have made chromosome biology a very important aspect, especially of clinical cytogenetics. As you would see, this is an example. This is from your lab, from Dr. Amit Ray's work. This shows fluorescence and fish. You can see the centromeric markers here. It is actually a slide from a Down syndrome child, which is appropriate for today, where you find that the chromosome is actually this, it has only 46 chromosomes, and yet it shows because there is a translocation of a chromosome on chromosome 15. So you find that there are two chromosome 15s. These are two chromosome number 21, this and this. Then you find a two chromosome 15, one here. Oh, sorry. And here, and then you will find one more chromosome 15, which is a thick blob here, which is because of chromosome 14, sorry, which has got chromosome number 21 here. So using fluorescence in situ hybridization, you can have elegant informations. These are again informations that you can get from meiotic cells, where you can use what you call the immunocytochemistry. You can use a marker, an antibody against, for example, a crossing over marker like MLH, and you can locate the chromosomes here. Here you will find that you can look what is called a spectral karyotyping, or where each chromosome can have a different kind of a color, so that if there are chromosomal rearrangements, as you can see here, you can find different markers to show you chromosomal rearrangements. So technology-wise, in last 40, 50 years, cytogenetics has developed enormously, as has developed microscopy. Got microscopes like focal microscope, you've got tunnel microscopy, and so on. And you can do such things as, now you can see this picture is where Dr. Sharma himself is doing microdissection of chromosomes. So he did microdissection in order to be able to dissect heterochromatin from his chromosomes, as I'll talk perhaps later, but at some point of time. And so, and this is a slide which again comes from this lab work of Dr. Amit Rai and Dr. Akhtar Ali, where you find a patient here with cleft lip and palate, but also other abnormalities, and you find that this has a supernumerary chromosome here, the bottom. Now, what is this chromosome is the question. And since it is not showing any banding, it's not easy for you to find out what this chromosome is. So you will use CGH microarray. So now microarray technology, and you can see if the two chromosomes are equally distributed, you will see in middle line, you will see it like this. But if there is a trisomy or a part of a chromosome which is more, then you will find this is gone up here, as you can see here. So there is a partial trisomy detected by CGH microarray. Now, what I mean to say is that now there are technologies, and there are therefore wide variety of uses made everywhere as well as here. And you can see how from this chromosome, this microscope, to now you have the confocal microscope. Same mitosis that you see Fleming, you can now see through confocal microscopy. And what you would find is that there isn't much difference between what was done by Fleming in 1882 and what you can get by confocal microscopy today. That means how accurate was the drawing. That is the key, friends. Not so much the microscope as the eyes and the mind. As somebody was telling, brain and heart, I think both the things have to come together to do good science. Now, I'll briefly touch upon the scenario in India. When I was trying to figure out, make out this talk, I very sincerely tried to find out as to whether in between, say, up to 1950, what kind of animal cytogenetics was being done in India. I must admit that it was not possible for me to find many reports. I could find 3 papers of Professor A.B. Mishra, who was the head of the department here, but those 3 papers were on particular chromosomes, but that was work done in Makino's lab in Japan in 1933-34. But there are some very interesting papers from Professor B.R. Shishachar on amphibian chromosomes. Again, they were made from sections. And I'm not going to show you those pictures because I don't have it readily available to me. But later workers who have looked at air-dried preparation of chromosomes of amphibians or frogs particularly, they find that what was reported in 1937 by B.R. Shishachar was pretty much the same as was seen by the people later on. So there are really not many good works on chromosomes, and I get it. Also want to share with you, when Professor Rai Chaudhary was then he was a PhD student, did PhD with Muller in Institute of Animal Genetics. And by 1940, he had submitted his thesis and he was on way to India. World War had already broken. So his ship was delayed. P.C. Kohler, who was also there at that time, the one who showed 2 and 48 of human chromosomes, and he said, Rai Chaudhary, you'll find it very difficult to do Drosophila genetics in India. Why don't you learn some chromosome work? And so Rai Chaudhary picked up squash preparation of grasshopper testis from P.C. Kohler. When he came back to India, that is what he started. So at least there are few people who came around the same time, who started the work around the same time. M.D. Srivastava in Allahabad University, Rai Chaudhary in Calcutta University, B.R. Sheshachar and Rajshekar Shetty in Mysore University, and G.P. Sharma and his colleagues in Department of Zoology of Punjab University. I must mention that cytology was not that bad in the earlier years. There were people like Professor Vishwanath and also Dr. Sheshachar, also like Dr. More. These people were good extranuclear cytologists, but they had not done chromosome work, which was started. Now, so these people, Rai Chaudhary, having been trained in radiation genetics under Muller, used radiation-induced chromosome aberrations and produced a very beautiful piece of work during those years. As luck would have it, in 1952, he was visited by his PhD examiner, C.V.S. Haldane, to whom he showed his chromosome work on grasshopper testis, of which he was admiring. But then he said, why aren't you doing genetics? And he said, doing genetics is not possible. I don't have an air-conditioned room. I need air because temperature is to be maintained. And that's how Haldane went back, wrote to the Prime Minister of India at that time. He said, I want to do Drosophila genetics. And a genetics lab came up in Calcutta University in 1954-55, and that's how Drosophila genetics started there. But prior to that, a lot of work was being done on grasshopper, radiation work, and on other insects on doing their cytotaxonomy. And some very beautiful piece of work around the same time, I must clarify, from Punjab University, from M.D. Chaucer's lab in Allahabad University. Actually, in MDS lab, there were some very beautiful insect chromosomes and XX chromosome mechanisms were shown, which is also shown by Rai Chaudhary and others from Calcutta. So, but after this, in 60s and onwards, we have had quite a bit of activity using cytogenetic tools in Indian laboratories. For example, Dr. A. Mukherjee, he came back from California, and then having spent a bit of time with Wehrmann and Tübingen, he came back to India and started his lab in Calcutta University. He used 14 chromosomes. Later, one of his students also was a faculty there, R. Chatterjee. In Kalyani, there was Professor G.K. Manna, who had a very large group of students from his group who worked on fish, on almost every other vertebrate, from fish to amphibians to reptiles and everything else. And his group spread out also in Odisha. So many of the universities in Odisha had a group of people, and they also had a cytogeneticist, Dr. C.Das, who was a product of Allahabad University, from Inder Gadi, who was working there. Delhi University had Dr. S.R.V. Rao, whose student was Dr. Thelma. You had seen, you have heard her. He was a remarkable cytogeneticist. For a short period of time, Dr. Lakhotia also worked with him as a postdoc, but he was, again, mammalian cytogeneticist and did beautiful piece of work, especially on Nasocchia, a rodent. In Mysore University, Rajshekar Shetty had a student in N.B. Krishnamurthy and Ashwatha Narayan. N.B. Krishnamurthy again worked on population genetics of Drosophila, just as some people were doing in BHU, as we shall see here. So a good number of people in Banaras University, Professor Rai Chaudhary would come, T. Sharma, S.C. Lakhotia, B.N. Singh, Mercy Raman, R. Raman, and their students and so on. They also began to work, did a fair bit of work in there. In IISc Bangalore, H. Rajchandra started to do some work on cytogenetics of insects, particularly silkworm. And in Allahabad University, again, Dr. M.D.L's student, Dr. Deepika Srivastava or Dr. V.S. Bhatnagar, they worked on bird chromosomes and on mammalian chromosomes and so on. So I would very briefly, and I should put a caveat to what I'm going to say now. it is not a comprehensive list of a very large number of people who have been working. I would have definitely missed a number of people. And I'm sure that I would have missed also some of the very important work that some people would have done. I have only tried to summarize some of the work that of which I was in knowledge, and I have then tried to gain knowledge in the last few days. So it's quite possible that I may have missed some very important discoveries or important researches made, but some of the major contributions that were made were here. Like, for example, in Drosophila dosage compensation, Mukherjee, of course, to begin with Beerman in Tübingen, but having come back to India, he developed that work very extensively, and he was very highly respected for this work because he showed that dosage compensation, which means that although the Y chromosome, there is only one X chromosome in male, two X chromosomes in females, but the X chromosome products of both the sexes are same. And this he showed was possible because X chromosome in male produced twice as much product as that a single X chromosome in female. So that was hyperactivity of X chromosome in male was a wonderful demonstration of dosage compensation. Then Sharchandra in IISc and one of his postdocs, Vani Brahmachari, who was later on in Ambedkar, with whom I think Richa has worked for some time. They worked on genomic imprinting because this silkworm shows a phenomenon where the paternal chromosomes get heterochromatized and eliminated. So how does a cell recognize that only it will eliminate only the paternal chromosomes and not the maternal? What is interesting is that what is paternal in one generation, what is maternal in one generation can in the next generation become paternal if it goes to the son. And the same chromosome which was maternal or that of grandmother is now recognized as that of the father. How does this mechanism take place is very beautifully illustrated by Sharchandra to begin with, but by Vani Brahmachari as in Ambedkar Institute. P. Sharma showed, as Mercy was talking about, that differential radiosensitivity between sexes, and there was a volume of work on radiation work was done. All of it we are not mentioning here. But the other work which gained some traction was the demonstration that a part of the chromatin, which is supposed to be junk within inverted commas, and we don't talk of it junk anymore, actually could be a driver of evolutionary process, as shown by Professor Sharma in Masduri complex, which is now called as Mastericola. And then there were examples of, for example, chromosomal rearrangements like inversion. Inversion polymorphism could play an important role in speciation in Drosophila, as it's been shown by from Banaras University by Dr. B.N. Singh and his group. But a very interesting piece of observation was made, actually experimentation was done by Dr. Ranganath in Mysore University, where he could show rapid evolution in the lab, creating cytoraces by varying the number of B chromosomes or supernumeric chromosomes, as we call them. This would give you some idea. This is the slide which shows you differential radiosensitivity. You can see the first. This is male sensitivity compared to the female sensitivity. You can see female are much less sensitive than males. This is the paper of Sharma and Das, which was published in Nature in 1981. Das and Sharma, so on. Sorry. These are some of the chromosomal rearrangements which take place. You can see there are chromatid aberrations when one chromatid is broken. This is a very nice picture. And there could be chromosomal rearrangements where both the arms of a chromosome get damaged and then they can make a dicentric chromosome. This is the picture of the heterochromatin's role in evolution, where you can see that this is from Mastericola. You can find that the X and Y are large, having the large heterochromatin, short arm in the whole of Y. But they also have heterochromatin in certain autosomal chromosomes, like 3 chromosomes here, 2 chromosomes here, and none here. And you would find that all these 3 are reproductively isolated. What we mean by reproductively isolated is that they are already incipient species. So the role of heterochromatin in evolution, or this is an example from Drosophila ananasi, where there are 3 very well worked out inversions, alpha, delta, and eta. This again comes from Dr. B.N. Singh's lab, where they show how these inversions and distribution in populations are population specific, which also suggests that they could have a role in speciation and differentiation. Then there are again on the functional aspect of cytogenetics, which was shown by Lakhotia and earlier also by Mukherjee, how gene function takes place. It was shown that certain stress like heat shock can modulate gene expression in particular ways. Only certain puffs will emerge when the shock is given. One of the such genes was, one of the puffs was what's called as 93B puff, which was not only expressed as a heat shock response, but also by certain other treatments like benzamide, colchicine, and other things. It was worked upon later on, and it turns out that this is actually speaking one of the first examples of long non-coding RNA. It is not often mentioned because we have got so many more examples of this, H19, and so on and so forth. But this is also one of the few examples of long non-coding RNA, which came up almost in 1990s. And then a very good example was work was done on snake sex chromosomes. Unlike mammals, where you find that the Y is very small and the X is quite big, you find in snakes the the X and the Y, which we call as Z and W in that case, are not that different in size. Actually, some of them are morphologically similar, and therefore it was assumed that very often the sex chromosomes they are made of because of the inversions. Lalji Singh in his work with Professor Raichaudhuri and Dr. Sharma showed that actually it is because of the heterochromatinization of the chromosome. So heterochromatin was playing a role in the evolution of sex chromosomes, and they showed very beautifully that this heterochromatin forms in female, unlike sex chromatin in males. In mammals, in snakes, females have a chromatin body made up of the W chromosome. That's why it's called as W chromatin. Similarly, beta heterochromatin, of course, this work was not done in India. It was done in Edinburgh, but it showed in that under electron microscopy how the beta heterochromatin is transcriptionally active, which means that the heterochromatin is not all junk, it could be functionally active. And similarly, certain interesting work was done by Professor Rao on the Soka sex chromosomes, which showed fragile X, which is, so you can see some examples here. You can see here, these are W chromosomes, and you will find that in many cases, W and this is Z and this is W, they're not very different in size. And that almost in different species you find that makes a W chromatin. This is the work that was done. Now this W chromosome, as you know, was later, particularly from Bungarus surrealis, which means crate, was used in finding out the W specific DNA, which is later on called as BKM, Bungarus banded crate minor satellite. And this was actually one of these molecules or sequence, GATA, which was used for DNA fingerprinting. In today's time, when we talk Lalji Singh as the father of DNA fingerprinting, it is this GATA sequence which came out from this BKM from Bungarus surrealis, from banded crate, that showed that. This slide I am putting again in order to show you how HSR omega, which is a product of 93D, shows, you can see coming out as a puff from a heat shock puff. Here you can see this comes out from an cement, and then it shows in different species you find this conserved. But what was curious and very important was that it did not make an RNA, a protein product. This work by Lakhotia and Mukherjee on the products of heat shock proteins showed that 93T did not have a product, though it was abundant in RNA. And of course, now we know much more functions that is associated with this. Well, one more aspect of this comes out, clinical cytogenetics, and this was the workshop held in 1962. It's a very interesting picture. It was organized by WHO, and it was conducted by Harold Klinger. And you can identify a few people: Dr. Sharma here, Dr. S.R.V. Rao here, Dr. N.G. Dev, who was the Tata Memorial, Tata Cancer Research Center of, who became director there, Dr. Dave, who was at AIMS. These people, this was the first time when lymphocyte culturing was demonstrated and people were trained. And many of these then in their respective places started lymphocyte culture as a mechanism to understand human cytogenetics. And these are some of the labs which are doing cytogenetics. I.C. Verma in Department of Pediatrics to begin with in AIMS. Then he retired He still works in Sir Gangaram Hospital. Kiran Kucheria in Department of Anatomy. Now there is one Dr. She now retired. There is one Dr. Mandal, Ashutosh Mandal, who looks after it. Manorma Thomas, she also is retired from St. John's, but she also started a very productive human cytogenetics lab, clinical cytogenetics lab. S.S. Agrawal, no more. Again, he started to begin with in KG Medical College and later on in SDPGI. Now that lab is now headed by Shubha Patke in SD. And they are all very nice. Dr. R.N.K. Bamezai started a laboratory in anatomy here in AIMS, in IMS, then moved to JNU and started a human cytogenetics lab there. Dr. Thelma started a fragile site diagnostic unit in Delhi University in South Campus. Dr. Chandak in CCMB as a big lab, diagnostic lab. Dr. Ahuja and then his student was forgetting his Kurtulan, what was her second name? I don't remember. So they made their cytogenetics lab. One lab you see here also, Center for Genetic Disorders. And there would be some more. I must admit, I may have missed some. But the important point is that a fair amount of work has now come about, and number of laboratories have come. I'm talking of cytogenetics laboratories. There are also large number of genetics laboratories which look at genetic disorders. I'm only confining myself to this. Similarly, there are very strong groups of cancer genetics, cytogenetics, and you will see Tata Memorial Hospital, ECTREC, ICPO, CDFT, all these places are doing very nice work. And you can see how using these technologies, you can see we have shown about aneuploidy, Down syndrome, Turner syndrome. Now you can see even a small deletion of a band can lead to a disorder. This again from Dr. Amit Rai's lab from here, CGD. And this is leading to von Wilms tumor. You can again see this is from Angelman, and you can find that there's only a small deletion, which you see it is deleted here. It is present this thin band that you can see, and that causes, if it comes from the mother, it causes Angelman. If it comes from the father, it causes Prader-Willi. That's therefore an example of genomic imprinting as a disorder. You can see Fanconi's anemia, a rare disorder, but if you give an insult like or a challenge like vitamins C, you can see what amount of chromosomal rearrangements it can generate. You can especially see chromatin rearrangements and exchanges. And as I showed you earlier, how CGH microarray can become effective in diagnosing the disorders. This is the last slide to show you how chromosomal rearrangements occur in cancers. As you remember, this was the thing that Theodore Boveri said, that chromosomal rearrangements are at the base of most of the malignancy, particularly translocations. If you remember, I also showed a slide where I showed how Dowell and Hungerford showed Philadelphia chromosome. There is a chromosomal rearrangement between chromosome number 9 and 22. This is this tiny bit of a chromosome we call as Philadelphia chromosome, because the Fox Chase Hospital, where this was discovered by Dowell Hungerford, and Dowell and Hungerford is in Philadelphia. Around the same time, it was also discovered in Buffalo in Roswell Park Memorial. They were a little late, thank God, otherwise we would have to call it as Buffalo chromosome. If you don't do anymore, we call it only Philadelphia. So, and there are techniques, you can see microarray, you can see, look at this slide. This is how enormous amount of chromosomal rearrangement takes place in cancers. And that's why as it aggravates, how it becomes so difficult for it to be treated, because how many different chromosomes, I mean, when I say different, understand this, one color is one chromosome. Now you see every chromosome here, or more than 50% chromosomes here have got more than one color, which means they have got translocation, and so many translocations. So this is how the science has grown up. Good thing is that India, for the first 50 years, perhaps we did not do much work. India was not a free country. But in the last 50 or 70 years since, I think we have been doing much. But that much is not enough. See, if we can print out names on one page of a PowerPoint slide, it doesn't speak good of a big country. I think we need many, many more people to be working, but the need is more. The cases are coming up, and we don't have people to take care of. If we are able to do that, I think people like Professor Tikaram Sharma would have been happy up in the heavens, and we here on the ground floor. Thank you very much. I hope you understand this picture is of the DNA material. This is nano DNA fibers. This is typical DNA nanomaterial. So when I say use DNA as a nano material, this is an example. This is made up of DNA. Thank you.
Thank you, sir, for showing us the grand history of cytogenetics with so many historic, elegant pictures of chromosomes and RNA starting from 1880s till date, and how cytogenetics was developed using so many varied models like pea, fish, salamander, Drosophila, sea urchin, insects, etc. It is very fascinating how genetics that started from term factor have traveled through chromosomes till genes. Thank you once again, sir, for teaching us that both brain and heart shall come together to do good science. I request our director, Professor A.Tripathi, to kindly felicitate Professor Raman. We will now take a break for refreshments. Please be seated. The break will be followed by playing few videos and audio clippings by Professor Sharma's student and vote of thanks. During refreshment, we will also present you all the dignitaries with small token of love, which are prepared from very talented kids from DEVA International Society for Child Care from CGD.
Sir, it is very nice to hear you from Delhi. Very happy to see the old chromosome pictures.
If anyone from the online audience would like to share their thoughts, we welcome them, please.
Yes, it was really wonderful hearing, sir. Are you able to hear?
Yes, sir, you are.
Are you able to hear?
Yes, sir, you are audible, sir. We can hear
Yes.
you, sir.
Yes, it was most wonderful hearing Professor Raman and seeing the beautiful pictures. The old pictures that nowadays we do not see. So very, very happy seeing them.
Thank you, sir. Thank you, sir, for your thoughts.
Thank you. अब कुछ बोलना है?
Hello. Yeah. We would like to have your kind attention. Meanwhile, we'll be playing the videos and audio clippings as well. So please enjoy.
Greetings to
Hello? Hello?
everyone who are attending Professor Tikaram Sharma's memorial lecture. My name is A.K.Tripathi Sharma. I was mentored by Professor Tikaram Sharma from the childhood till his last days. I could contribute with guidance from Tikaram Sharma locally, nationally, regionally, and globally. I have been able to serve as Vice Chancellor of Perry School of Advanced Studies in New Delhi, Deputy Director General of International Center for Integrated Mountain Development, ICMOD, which is an intergovernmental organization of 8 countries in the Hindu Kush Himalayan region. And currently, I am the chairperson of the Scientific Advisory Committee of the G.B. Pant National Institute of Himalayan Environment under the Ministry of Environment, Forest and Climate Change.
I completed my PhD with Professor Sharma in 1988, working on systematic exchanges in diamonds, mainly moonshine and human. After that, subsequently I joined CCNB, retired from there, and I'm now continuing as advisor research to Osmania University affiliated to the genetics department. I would like to say that the work I did with Sir and Sir's passion for finding functions for heterochromatin actually laid the foundation for my work at CCNB, wherein we went on to discover non-coding RNA-mediated regulatory functions of Y chromosome heterochromatin in man and mouse.
Good afternoon, everybody. I am Bhudev.C.Das. I did my PhD with Professor T. Sharma in 1976 to 1982. And then I joined ICMR. I must thank all the students, the post-graduation, graduation, and PhD students who have taken this initiative to start the Professor T. Sharma Memorial Lecture Series. And it is rightly selected that the first student of Professor Sharma, Professor Rajiva Raman, is speaking today. I spent whole life in ICMR, and I retired as director from ICMR in 2008. And from there, again, I joined Delhi University, Dr. B.R. Ambedkar Center for Biomedical Research as director in 2008 itself, and then continued till 2014, and then ultimately resigned from ultimately I retired from there, and then 2014 itself I joined as chairman of molecular medicine and stem cell research institute at University. I created this institute from the scratch, which has Clarice to a full-fledged institute with 16 faculty and several PhD and university programs in different discipline frontier areas of research. I also have some other responsibility as the Dean of Health and Dean of Health and Allied Sciences, as well as the Chairman of University Research Council, and also Vice President of MIT Science, Technology and Innovation Foundation. Thank you very much for your presence, and this is one slide, just you can see it. Thank you.
Professor Sharma was one of the few teachers who could keep the class awake by his sheer enthusiasm. I still remember his classes where he taught us chromatin organization, jumping from one end of the longboard to the other. His passion for these topics exuded out. He had a flair for drama. His hand movements while teaching us how homologous chromosomes with translocations or inversions segregated out during meiosis was mesmerizing. It was fun attending his classes, and those images still remain etched in my mind. I'm sure he's keeping everyone on their feet or toes wherever he is now. This is Pradeep, one of his MSc students from years back.
Hello everyone, I am Rakesh Tamang, Assistant Professor at the Department of Zoology, University of Calcutta. First of all, I'd like to thank the organizers for conducting such a wonderful tribute to our beloved late Professor Tikaram Sharma. And I feel very fortunate enough to talk about him in this event. My interaction with Sir started way back when I was in class 8. He visited our school and talked about his journey from a humble boy to a world-famous scientist. That speech inspired many students, and I was one of them. Sir knew about the challenges of our place, Darjeeling. He had a vision of building a university, a research institute, and a good healthcare system in our place. Now, it's our turn to carry forward his vision. Thank you very much.
Professor Sharma had a passion for teaching. We had to be on our toes during his class, as he exactly knew we were not paying full attention. I still remember the topic on transversion, deletion, duplication, and his hand movements at that time. At that time, when he said he picked up a book on microscopy from the airport, it was difficult to understand or rather digest. I will always remain thankful to him for stepping in for me at a crucial junction. I joined as an RA just after my Viva Voce examination. I'll remain extremely thankful to him for this. Thank you very much. This is Shweta Saran from School of Life Sciences, Jawaharlal Nehru University.
From Tulsi. I joined the Center of Life Sciences under North Bengal University under the able guidance of Professor T. Sharma back in '78 for my PhD program. I was very closely associated with him and am indebted to him for shaping my life and making me a better version of myself. Professor Sharma was a visionary and a true teacher. His energy was motivating. I marvel at his passion for innovation even in his later years. His dream to set up a satellite genome center in Cheong remains a dream, unfortunately, with his untimely departure. It gives me immense pleasure and pride to think he was my mentor. I am privileged to have learned a lot from him. One day, I hope I can be as inspirational to others as you were to me, dear sir.
Myself, Mr. Satyendra Moitti, a retired associate professor in zoology from the Department of Zoology, APC College, New Veracruz, gratefully indebted to late Professor T. Sharma to have an opportunity to work with such a enthusiastic, knowledgeable teacher. And I am also to mention my immense indebtedness to Professor Rajiva Raman, Department of Zoology, BHU, the then postdoctoral research scholar with late Professor Dr. T. Sharma, helping me in all respect, including the research, with all his charming disposition and love. It appears we showed the seed of human cytogenetics that remained dormant for some time, followed by its germination being nurtured by the now National Professor Dr. Rajiva Raman, and has grown up into a huge tree having colorful flowers, yielding fragrance both nationally and internationally, for which full credit should be endowed to Professor Parimal Das, the present incumbent of this Institute of Human Congenital Disorders. Of course, Professor Rajiva Raman, his PhD guide, was at the helm. I wholeheartedly pray its more and more ascendance to help mankind in future. Thank you all for giving patience.
Hello everyone, I am Dr. Sunita Singh. At present, I am working as professor in Department of Zoology, MMBPHU. I joined Professor T. Sharma as a PhD student in the year 1989 and completed my PhD in 1996. He was a brilliant teacher, and above all, he was a wonderful human being. He was a visionary and way ahead of his time. He was like a father figure for me. I was lucky to have him as my guide and mentor. Though he is not there with us, but I know his blessings are always with us. Today is his birthday. A very happy birthday to you, sir.
Good afternoon, everybody. I am Jayashree Mohanty, completed my PhD in 1992 under the guidance of Dr. Rajiva Raman. The very day I joined our lab, I came to know that my sir is also there. And when I went to meet him, the first question he asked me, heterochromatin kya hai, janta hai? And that was so impressive that when after a long time I joined my job, I also asked the same to my undergraduate students. His dynamic, jovial and inspiring personality can never be forgotten. When I changed my PhD topic, he said me, "Man se kaam karte raho. Your aspiration must be fulfilled." And I have been following those words since then. Wherever you may be, sir, we need your blessings always.